Hi everyone, this video will provide a big picture overview of metabolism. We'll talk about two things. First, what is metabolism? And then what can go wrong during metabolic deficiencies? So metabolism is the set of reactions within us required to maintain life. And it's how we convert the foods we eat into ATP, a usable form of energy, into all the molecules that our cells and tissues need, and into the waste products for excretion. So we'll talk about these one at a time. First, how we convert the foods we need into usable energy, or ATP. The key thing here is that we oxidize the carbons from the fats, carbohydrates, proteins, and if we consume ethanol, from ethanol, to make ATP, as well as the waste products of that oxidation process, CO2 and water. And oxidation is the loss of electrons or hydrogens, or also the gain of oxygen. But often during metabolism, it's really the loss of the electrons or hydrogens that's important for the oxidation. And the enzymes that catalyze this process are dehydrogenases. So I just want you to remember to look out for dehydrogenases as being key enzymes that catalyze these oxidation and reduction reactions. So let's look at an example. This is the pathway of fatty acid oxidation, or beta oxidation. And you'll see that there are two dehydrogenases, two oxidation steps in this process. So what we're looking at the top is just a piece of a long chain fatty acid. This R group represents a long chain fatty acid tail here. And it's called beta oxidation because you're going to see the beta carbon here is the one that's getting oxidized. So the first step is catalyzed by acyl-CoA dehydrogenase. This is actually a family of different dehydrogenases for different length chains of fatty acids. Right? This is a dehydrogenase that uses FAD cofactor. So as the fatty acid is getting oxidized, we see that by the double bond and the loss of a hydrogen, the FAD is getting reduced. The second step, water gets added, and that actually provides the oxygen atom that's now on the beta carbon. The third step is catalyzed by another dehydrogenase. It's another oxidation step. And this dehydrogenase uses NAD as a cofactor. And now we see that the beta carbon has been essentially fully oxidized to a ketone. In the last step, coenzyme A is activated to cleave off an acetyl-CoA, we see that here, and the fatty acid attached to coenzyme A that's 2 carbons shorter. So next, these 2 carbons of acetyl-CoA will be fully oxidized to produce CO2 and water, and actually CO2, I should say, in the TCA cycle. So let's review that next. Okay, so here we have acetyl-CoA in the TCA cycle. This is happening in the mitochondrial matrix, same place that the fatty acid oxidation occurred. The first step is the acetyl-CoA is put on the 4-carbon oxaloacetate by citrate synthase to make citrate. Then the citrate gets isomerized to isocitrate. Then we have another dehydrogenase that produces NADH. A CO2 comes off, so we go from 6-carbon isocitrate to 5-carbon alpha-ketoglutarate. And we have another dehydrogenase, alpha-ketoglutarate dehydrogenase, which releases both a CO2 as well as producing an NADH. And we have succinyl-CoA. The next step, we synthesize a GTP as the coenzyme A is released to produce the 4-carbon succinate, which then gets reduced to fumarate. The fumarate gets rearranged to malate, and the malate gets oxidized by malate dehydrogenase to get us back to oxaloacetate. So in the end of this whole process, we've oxidized two carbons. Two carbons came in as acetyl-CoA. We've lost two carbons here. We have the same number of molecules of oxaloacetate as we started with, so no gain or loss there. But we have gained 3 NADHs, an FADH2, and a GTP, which is energetically equivalent to an ATP. Okay. So that's what we end up with in the TCA cycle. So what happens with all that NADH and FADH2? Okay. The CO2 we exhale, this is where most of the CO2 that we're exhaling comes from. The NADH and the FADH2 will now fuel the electron transport chain or the respiratory chain to produce most of the ATP. Okay, so this is an image of the respiratory chain, electron transport chain. It occurs in the inner mitochondrial membrane. We have lots of protein complexes that catalyze these electron transport reactions. The first one, complex 1, is also called NADH dehydrogenase because it's the enzyme that takes the electrons from NADH and the hydrogens and produces NAD+, as we have loss of electrons. So the NADH is getting oxidized here. The electrons are getting transported through, and energy from the electrons is used to pump protons. Approximately 4 protons are pumped per pair of electrons entering complex 1. Complex 2 is also succinate dehydrogenase, so one of the enzymes of the TCA cycle, the exact same enzyme. The FADH2 that was produced in the TCA cycle is reoxidized to FAD, and the electrons are transferred to coenzyme Q, which is a lipid that floats around in the inner membrane here. Coenzyme Q gives those electrons to complex 3 that uses the energy from those electrons to pump 4 more protons into the inner membrane space. Complex 3 gives those electrons to cytochrome C, a small protein that donates those electrons now to the terminal complex in the electron transport chain, which is complex 4, which pumps some protons, and those electrons and hydrogens are given now to oxygen, the ultimate oxidizing agent, which has a very high affinity for electrons, which is what drives energetically this entire process. We produce water here in complex 4, and we have this proton gradient. So this electrical chemical gradient, which is a lot of energy. So this high energy gradient now can fuel this little rotary motor, complex 4, also called ATP synthase, which puts ADP and phosphate together to make ATP. The vast majority of ATP that's produced in our body is produced here at ATP synthase, or complex 5 of the respiratory chain. So in this process, we've gone from oxidizing carbons to the NADH, FADH2, which fuels the electron transport chain so that ATP synthase can make ATP. And then on the final part of this slide, just wanted to show you about how many different proteins are involved in each one of these complexes. So each one of these ovals at the bottom represents a protein. These are complicated machines, these electron transport proteins, and they're encoded both by mitochondrial genes, outlined in red, and nuclear genes. Okay, so for complex 1, complex 2 is all nuclearly encoded. Complex 3 has 2 mitochondrially encoded genes, 4 has 3 mitochondrially encoded genes, and complex 5 have 2. And the reason I'm showing you this is that there are people who inherit mutations in the mitochondrial genome. Every gene on the mitochondrial genome that encodes a protein encodes a protein in the electron transport chain, or ATP synthase, so in the respiratory chain, right? So, and there's also lots of nuclearly encoded enzymes in this process. So if you have a mutation in the mitochondria, if it affects you, it's because it affects the electron transport chain, or ATP synthase. You can also have a mutation in nuclearly encoded genes that also affect these processes. Okay, so that's fatty acid oxidation, which I'm simplifying here, to the TCA cycle, to the electron transport chain and ATP synthase. All right, so we can also oxidize glucose, and I'm showing here glucose and simplified the process of glycolysis to pyruvate. Pyruvate can be oxidized by pyruvate dehydrogenase to make acetyl-CoA. We produce NADH in the process and lose a carbon dioxide, right? So this is also happening in the mitochondria, and the acetyl-CoA, those two carbons, get oxidized through the TCA cycle in the electron transport chain as well. Also, the amino acids from our proteins can also be oxidized in this process, and each protein enters the TCA cycle at a slightly different place. And finally, if we consume ethanol, 2-carbon ethanol gets oxidized, those carbons, to acetyl-CoA, producing NADH, primarily in hepatocytes. So we can make lots of NADH just by the process of oxidizing acetyl-CoA, oxidizing ethanol, and then the acetyl-CoA can enter the TCA cycle. Okay, so that's how we make ATP. The second thing in metabolism is, how do we make all the molecules that our cells and tissues need, like the nucleotides, like cholesterol and the steroid hormones? We have pathways to do that. All the non-essential amino acids, glycogen, so storage form of sugar, and all needed sugars. You may recall that there are no essential sugars. There are no sugars that we have to get from our diet. We can synthesize every sugar that we need in the body. And remember that there are lots of glycolipids and glycoproteins that use all kinds of sugars on them, but we can make all of those sugars. We have pathways to do that. We have to make triacylglycerol for storage of fats, and phospholipids for membranes. We can do that. And we make heme. We make heme from amino acid and TCA cycle intermediate. And we have to make all our neurotransmitters, such as dopa, dopamine, norepinephrine, epinephrine, and serotonin. And so you'll find these pathways are all on the Pathways of Human Metabolism metabolic map. And the third thing is, we need to make waste products for excretion. And we'll just look at 2 of those here. One is urea. So urea is this simple little molecule, 1 carbon, 2 amino groups, and an oxygen. And urea is the waste product from catabolizing amino acids, right? And I just want to contrast that with uric acid. So this is uric acid. It is produced by catabolizing purines. We excrete both of these in the urine, which is where the UR comes from. And I know sometimes students mix up urea and uric acid. So again, urea is the waste product from catabolizing amino acids. Uric acid is the waste product from catabolizing purine nucleotides. All right, so let's finish up by talking about what can go wrong during metabolic deficiencies. So we have inborn errors in metabolism, or genetic deficiencies, and they can either be complete or partial deficiency, meaning that the gene can have a mutation that means that there's no enzyme activity at all, or there could be partial enzyme activity. And that has, depending on the type of mutation, it means that either these problems can show up at birth or later in life, or maybe only when you have a particular metabolic stress. There's also vitamin and mineral deficiencies to be aware of. So Remember that all the B vitamins are enzyme cofactors, and many minerals are required at enzymes. So mineral deficiencies can show up as an enzyme deficiency as well. And basically, there are three general categories of things that can go wrong if there is a metabolic deficiency that results in pathology. So if we look at a generic metabolic pathway here from A to D, and there's a metabolic block that just represents a decreased enzyme activity compared to normal, right? We can either have the substrate accumulating, and sometimes that's toxic. If we have too much of A here accumulating, that can cause pathology itself. Or it could be that A gets converted to something else, some other metabolite that is toxic. Or it could be that we don't have enough of a needed product. So we have a deficiency of a product, and that can cause pathology. So let's look at some examples of that. Here's an example of Myophosphorylase deficiency, which is muscle glycogen phosphorylase deficiency. It's also called McArdle's disease. So a reminder, glycogen phosphorylase is the enzyme that activates phosphate to cleave off glucose molecules from glycogen, resulting in glucose 1-phosphate, which can then be isomerized to glucose 6-phosphate. So this phosphate gets moved up here to the 6th position, which can then enter glycolysis to form pyruvate. And depending on the situation in the muscle cell, if it's anaerobic, then the pyruvate will get converted to lactate to produce 2-ATP, or if there's plenty of oxygen available, the pyruvate will be fully oxidized to CO2 and water and lots of ATP, right? So if there is a deficiency in glycogen phosphorylase, the problem is that we have insufficient ATP for muscles, particularly during quick bursts of energy. And that is the pathology that results, is muscle weakness and soreness after exercise. Another example is phenylketonuria, or PKU. This is a disease that is caused by a deficiency of phenylalanine hydroxylase, which is required to metabolize phenylalanine to the amino acid tyrosine, shown here. But it can, and that metabolic block results in an increase of phenylalanine and its other metabolites, phenylpyruvate, phenylactate, and phenylacetate, all of which are toxic for the brain. Now, phenylalanine hydroxylase requires this, requires the cofactor tetrahydrobiopterin, which we require this enzyme to put it into the reduced form. And if this enzyme is blocked, is deficient, then also PKU can form. And we have a buildup of these metabolites and brain toxicity. All right, so to summarize, metabolism is the reactions needed for life. We use these reactions to extract energy and make ATP by oxidizing carbons from our foods, to make all the molecules that the body needs, to make excretable waste products. And in medical sort of metabolism, we're concerned about metabolic deficiencies that can arise due to a decrease in production of needed molecules and or the buildup of toxic compounds. And that ends our video.